The topic for today's discussion is about drug development and regulation. In this, we will talk about the clinical trials, animal testing, applications like NDA, IND, patent right, generic, in vitro studies, and etc. So we'll talk about other things in our today's discussion. So for that, what I need your concentration. So what you need to do is concentrate while I'm explaining the lecture. This lecture is going to be explained in very simple words. So just concentrate, okay? So let's start our discussion from the very first point, that is the drug development and regulation. So now, what we do actually is when the drug is supposed to be developed and then regulated, for that the concentration needed is on the sale and the use of the drug. So for this, there are special agencies in several countries. So the book that I have just taken this lecture, not book, but actually books. So one of the books is actually Katzeng. In that, I got this important point, that is regarding the US. So we are talking about the US. Regarding US, there is an agency known as FDA, Food and Drug Development Authority, that is responsible to regulate the sale and the use of the drug. So what is then the procedure for that? If there is a drug supposed to be developed and regulated, what's supposed to happen? Simple. First of all, in vitro studies are supposed to be performed. Suppose we have a causative agent that is causing the disease. So we will take that agent, on that we will apply several number of chemical compounds. We will check which of the compounds is actually best to fight against this particular disease causing agent. So what we do is actually we take the biological and synthetic compounds, we apply all those compounds one by one, and what we get one of the compounds which is actually supposed to be effective. So like this, hundreds of compounds are actually tried. Means we try hundreds of compounds on this particular disease causing agent. So some of the compounds, they show action. So then we just take those some compounds like as leading compounds. Then from those hundreds of compounds, we take some leading compounds. Then those leading compounds are actually taken, then they are applied on the animals. So we do animal testing via leading compounds. In the animal testing, we check the efficacy and selectivity of these leading compounds. Then whatever is actually the leading compound, that one of those leading compounds, again we do a selection. We take very specific one. Then we just go for the application, that is known as IND. IND stands for Investigational New Drug Application. Now this is not just this IND, investigation new drug application. This is application that is supposed to be for the exemption. So that's why it is known as investigation new drug exemption application. Means it is a kind of application that is actually given to the FDA for what? For the allowance on the clinical trials. For the allowance for the clinical trials, that we want to do the clinical trials, means we want to do the experimental studies on human. So what happens is actually we take the application, we submit it to the FDA, then FDA allows us. So accordingly, then what we do clinical trials. In the clinical trials, we do experimental studies on humans. So what happens here, that leading compound, which was actually the very specific one, we take that. Then with the help of that, we do phase 1. In the phase 1, we take 20 to 100 individuals. On that, we just try that specific leading compound. So what happens? We take very minor dose. Okay, so we just want to check the safety and the pharmacokinetics of that particular drug that is supposed to be now discovered and supposed to be developed and regulated. So what we do then, we apply a very minor dose on in the phase 1 on 20 to 100 individuals. So a very low number of the healthy subjects are taken. Means a person not having a disease. We just take that person and just we put in all these. Means we take 20 to 100 individuals. That all of the individuals, they are actually disease free. Means they don't have any kind of disease. So what we are checking in this particular phase is actually the safety and the pharmacokinetics of that particular drug that we got from these trials, from the in vitro studies, from animal testing. So after these two steps, we got the leading compound. We just took that leading compound and then we tried that leading compound on individuals. Like we just try on 20 individuals or on 100 individuals. So a group must be within the 100 individuals. So all of these must be having a healthy state. They all must be healthy. So here we take healthy subjects. So what we do actually is we just observe safety and pharmacokinetics. Then in the phase 2, what we do is we take a little bit larger number, that we take 100 to 200 individuals. So now these individuals will be diseased individuals, means all the subjects will be having the particular disease against which we are supposed to apply this drug, this new compound, leading compound. So what we do here is actually we want to check whether this disease is supposed to be cured or not. I mean, what is happening with the diseased individuals if we just take these drugs? In short, we are checking the efficacy of the drug, that the drug which is now under our investigation, whether this drug is effective or not. We check efficacy in the second phase. Then you go for the phase 3. Here we increase the number, that is from 1,000 to 6,000, and again here the individuals are all unhealthy subjects. So again here we just check the further the efficacy, and here several studies are performed, double blind, etc. So we will just tell about those in research purpose. So here regarding this topic you must just concentrate is that regarding phase 1 we check the safety and efficacy, safety and pharmacokinetics. In phase 2 we check the efficacy, and in phase 3 we increase the number. And then after that when we go through all these phases, phase 1, phase 2 and phase 3, we again put all the data in an application called as NDA, new drug application. This is again provided to the FDA. Again FDA provides approval for this for the next step, that is phase 4. So we file NDA application, that is again submitted to the FDA. FDA approves it, and then we go for the phase 4. In the phase 4 what we do, we just take the drug and we put it in the market. Then again for about 20 to 40 years we do the checking of this particular drug that has been marketed, whether the drug is showing this the particular result that we got in the clinical trials, animal testing or in vitro studies. So now what is actually the drug doing actually in the market? We again do the cross check, that is called as post marketing surveillance. This takes for about, I think so far if I'm not wrong, phase 4, 15 to 20 years. So this is the duration that is actually for this particular drug, and the same goes for the patent. Now what is patent and generic? Simple. Now a drug that I have discovered, I am the owner, and it is my right to gain the profit or to go through the business point of view of this particular drug. So I have the right. That right is actually called as patent right, and I will be given this right by the FDA, and this right may include the duration, that is from 10 to 14 to 20 years. So it may exceed, so it is in short between 10 to 20 years. So this is the duration for this patent right. So in short what we say, that is regarding patent right, I will be given the authority to just produce the drug and to market the drug. No one else can synthesize the drug that I have for a specific duration. Now as the duration expires, then everyone can have application, and that application will be submitted to NDA, and NDA will give the approval. FDA and FDA. So in short, application at the end comes to the FDA. So then FDA allows the particular company that you can now produce more generic that you want. There is a kind of necessary thing, that is, if the compounds are bioequivalent, then that company will be allowed to market the generic product of its own company. So this is the patent and generic, the difference between the patent and generic. Patent is actually the right that is actually with the owner, okay? The right, that is with the owner, for how long the owner is actually synthesizing the drug and then marketing the drug. So after this duration that expires, then we say that patent is expired. Then all the other companies, they just try for the production. What they do, they provide the bioequivalency of the compound that they have, and then they just give to the NDA and FDA. So FDA approves that now from now onwards the other companies can also synthesize the generic, okay? So this is how the difference we get between the patent and generic. Now coming towards the next point, that is the exception. Now what is the exception? Regarding phases, phase 1, phase 2, phase 3 and phase 4, some drugs are there which are actually causing very life-threatening problems, like cancer, carcinogenic drugs. So we do what? We just take some different requirements are there for those kind of drugs. Sometimes some of the phases may be skipped, like phase 1 is already skipped because here we take healthy subjects. So in carcinogenic drugs we don't take healthy subjects, and some other requirements are also there for the carcinogenic drugs. So this is actually exception, okay? Exception that I told you right now, that different requirements are supposed to be required for the life-threatening drugs. We don't go through such kind of clinical trials and etc. So we then go through some other methods and may be required. So whatever is required, we'll go through for that. And regarding the cost, it actually costs $500 million approximately. This is the average value when there is the development of the new drug and regulation. So regarding the time point of view, in vitro studies takes place about 2 years, and animal testing again 2 years, clinical trials about 5 years, and whereas the fourth phase, it takes about 15 to 20 years. So in short, this is about the timing, the cost, exception, the phases, animal testing, in vitro studies, and all. If still you have confusion anywhere, feel free to ask us in the comment box, and thank you for watching.